So, we will now discuss about the tryptophan operon. Tryptophan is an essential aromatic amino acid and it is normally required for the synthesis of all proteins that contains tryptophan. So it is an essential amino acid and normally tryptophan is required for the synthesis of all the proteins that contains tryptophan. Now, if tryptophan is not present in the medium in adequate quantity, then the bacterial cell has to make tryptophan then, as it is needed for the growth of the bacteria, right? So that's why the tryptophan operon comes into play. The tryptophan operon in E. coli contains 5 structural genes naming as TRPE, TRPD, TRPC, TRPB, and TRPA. So these are the genes, the 5 structural genes. In case of lac operon, we have shown 3 genes right next to each other, coding for the enzymes that will deal with lactose. But in case of tryptophan operon, it contains 5 structural genes TRPE, D, C, B, and A. It also consists of some regulatory elements, mainly the promoter. The promoter is designated by the letter by the symbol TRPP. P is in capital. Then operator TRPO, the attenuator, which is a very important function, the attenuation, as we will see during the TRP operon, and secondary internal promoter known as TRPP2 and T terminator, which is designated by TRPT. So I will like to show you the TRP operon now. So this is the basic structure. Okay, so this is the main structure of TRP operon as shown in the top. From the left, if you see, there is TRPP, then comes TRPO, the attenuator TRPA, and then the 5 structural genes TRPE, TRPBD, TRPC, TRPB, and TRPA. Within the TRPD, there is also another regulatory sequence called TRPP2. We will discuss them one by one, what enzyme does they encode and what's their main function is. Okay, so this TRPP2, as we have seen in TRPD, it is a secondary internal promoter found in tryptophan operon. It is another promoter, the internal promoter found in tryptophan operon. But the main specific thing about the TRP operon is your attenuator, which is known as TRPA. What is attenuator actually? Attenuator normally, like regulation by the TRP operon, attenuation is a mechanism. It is a mechanism for reducing the expression of the tryptophan operon when the levels of tryptophan are high. Suppose the tryptophan levels are quite high, then the tryptophan operon won't need to make tryptophan. Bacteria don't need to make tryptophan now, so it will turn off or it will reduce the expression of the tryptophan operon when the levels of tryptophan are quite high. So they will not totally block the tryptophan operon. Rather than blocking the initiation of transcription. Attenuation is a process where it prevents the completion of the transcription of the genes associated with the tryptophan operon. So during regulation, for the regulation purpose, suppose there is tryptophan quite high found in the medium, so now this bacteria won't need these genes, these structural genes required to make tryptophan. So they will rather than they block this tryptophan operon, they will do attenuation. Attenuation is a process where it prevent the completion of the transcription of those genes, of those structural genes associated with the TRP operon. When the tryptophan levels are quite high, then attenuation will be very high because it will cause the RNA polymerase to stop prematurely. It will initiate the RNA polymerase to do, but it will stop the RNA polymerase prematurely when it is transcribing that TRP operon. So we will focus this attenuation at the lastly. First we have to discuss about what does tryptophan operon normally do, and the genes that are the structural genes that are associated with the tryptophan operon, what does they do, what are the functions of them. Obviously all of them make a single function, together they will make tryptophan. When the level of tryptophan will be low, then this tryptophan operon will be turned on, but if the level of tryptophan is quite high, then we don't need to make tryptophan. So by attenuation process, they prevent the completion of transcription. Thus, it will reduce the RNA polymerase. It will not reduce, it will cause the RNA polymerase to stop prematurely when it is transcribing the TRP operon. So the 5 structural genes that are found on tryptophan operon, as we will discuss one by one later in the next pages. These 5 structural genes of tryptophan operon code for only 3 enzymes. 5 structural genes code for 3 enzymes. 2 enzymes containing 2 different subunits that are made by 2 different genes. There are some enzyme which is made up of multiple polypeptide chain that is encoded by the TRP operon genes to make a complete whole enzyme, right? Okay, it means to say in the bracket that has been given that 5 structural gene coded for 3 enzyme. That means what? There must be some 2 enzymes which have 2 different subunits. These 2 different subunits encoded by different genes. These different gene, when they produce that polypeptide chain, this polypeptide chain will be further translated and they will bind to form a single functional enzyme. So these 3 enzymes are actually needed for the synthesis of tryptophan from chlorismate. Chlorismate is an important biochemical intermediate. As you can see here, it is written, it is an important biochemical intermediate found in plants and microbes, and it is the precursor of all aromatic amino acid. This chlorismate is the precursor for all aromatic amino acids like tryptophan. It is the precursor for tryptophan. It will make tryptophan further. It will make tyrosine and phenylalanine. We are discussing about from chlorismate, tryptophan will be made with the help of these 3 enzymes that are found in the tryptophan operon, and the 3 enzymes are coded by 5 genes, and the name of the 5 genes I have mentioned: TRPE, D, C, B and A. On the upstream, it also consists of promoter, it also consists of operator acting as a on off switch. As you can see here, it also consists of promoter and operator acting as an on off switch. It consists of an attenuator deals with attenuation, regulation of the TRP operon. When there is high level of tryptophan, we don't need this tryptophan operon to be opened at that time. So through attenuation, it actually prevents the completion of RNA polymerase transcription. Okay, there is also secondary internal promoter called TRPP2 found at the TRPD and a terminator called TRPT. So let's move to the next page. Okay, so it is saying this tryptophan repressor is always turned on. Repressor is another regulatory molecule which is formed somewhere in the upstream. This tryptophan repressor is always turned on unless it is repressed by a specific molecule called co-repressor. Tryptophan repressor who mainly regulates the TRP operon, this tryptophan repressor is always turned on unless it is repressed by a specific molecule called co-repressor. Tryptophan here acts as a co-repressor. Tryptophan normally, this tryptophan repressor always turned on. Okay, again I repeat, tryptophan repressor always turned on means it becomes active unless it is repressed, its activity is repressed by a co-repressor. So here tryptophan act as a co-repressor to shut down the synthesis of enzymes needed for the tryptophan biosynthesis. So here we can see in the figure tryptophan level is quite high, so it bound to the tryptophan of it bound to the repressor, tryptophan repressor, and this repressor actually change its shape and binds to the operator. The operator will prevent the RNA polymerase to bind and carry out the transcription of these genes that deals with formation of tryptophan. So the tryptophan operon is designed to make tryptophan. If it is not present, if bacteria cell don't get tryptophan, then a number of different genes called TRPE, are required to make that tryptophan. Okay, suppose there is high tryptophan concentration, as you can see in the figure. If there is high tryptophan concentration, then what will happen? This tryptophan is going to fit inside the repressor and going to change the shape of the repressor so that the repressor can fit into the operator. As I have already mentioned from the beginning, this operator will act as an on-off switch. Thus, the repressor then prevents the RNA polymerase to transcribe these TRP genes because there is sufficient TRP tryptophan. So they will won't let this TRP genes to be transcribed by RNA polymerase. Okay. So these are the first condition, that if tryptophan level is quite high, then your tryptophan itself act as a co-repressor. Tryptophan binds to the repressor protein, repressor change its shape, it binds to the operator, operator acts as an on-off switch, it turns off, so it prevents the RNA polymerase to bind and carry out the transcription of this genes that are needed for biosynthesis of tryptophan. This was in case of high tryptophan concentration. In case of low tryptophan concentration, what happened? The repressor changed its shape again because there is no tryptophan present or very low tryptophan present. So tryptophan would not able to bind with the repressor. So repressor changes its shape. It causes to remove from the operator, and thus the operator become turned on, and RNA polymerase interacts with the promoter and starts transcription of the TRP genes that are needed for the tryptophan biosynthesis. So it's just an opposite of lack operon, right? Here the repressor moves out from the operator to turn the gene on, right? To turn the gene on, to turn the tryptophan operon on. Thus RNA polymerase now interact with the promoter and starts transcription of the TRP genes which are needed for tryptophan biosynthesis. So this figure is about the tryptophan operon in the presence of low or no tryptophan in the medium, and thus easily the three enzymes that the genes will be transcribed by RNA polymerase because the repressor won't fit into the operator now because of low tryptophan concentration. It does not change its shape actually at this time. The repressor become inactive. So these are the two cases. One case, the first case when tryptophan is in high concentration, then it fit into the repressor protein. Repressor protein change its shape, bind to the operator, and the operator prevents the RNA polymerase to bind to the promoter and carries out the transcription of these genes. It prevents that. In case of low tryptophan concentration, what happened? The repressor change its shape and it moves away from the operator. The operator turned on. It allows the RNA polymerase to bind and carry out the transcription of these genes that are needed for the biosynthesis of tryptophan. Now, about the tryptophan repressor. This tryptophan repressor is a homodimer. It is a homodimer. Homo means similar, dimer means made up of two subunits. That means it contains two identical subunits. This tryptophan repressor can bind to the two molecules of tryptophan, which here acts as a co-repressor. Tryptophan repressor can bind with two molecules of tryptophan and then bind to the tryptophan operator to turn off the transcription, or you can say to turn off the TRP operon. So it is a dimer, right? Homodimer means two identical subunits it contain. And this tryptophan repressor can bind with two molecules of tryptophan, which will ultimately bind to the TRP operator to turn off the transcription of the TRP operon. So here we can see what it is written here. Here two polycistronic mRNAs are produced from the tryptophan operon. One polycistronic mRNA derived from all the 5 structural genes from E to A, from TRPE to TRPA, that one, that polycistronic mRNA, and another polycistronic mRNA obtained from the last 3 genes from the TRPC to TRPA. So two polycistronic mRNA will be produced from this tryptophan operon. One which will be derived from the all the 5 structural genes, that is from TRPE to TRPA, that one polycistronic mRNA, and the other one will be obtained from the last 3 genes. Now, what does each gene encode? I have not mentioned now. Okay, we will compare them one by one. First come your anthranilate synthetase. Anthranilate synthase, TRPE. TRPE encodes anthranilate synthase. Normally, this TRPE produce anthranilate from chlorimic acid, or you can say it produce anthranilate from chlorismate. Okay. Next is TRPD. TRPD cooperates with TRPE. TRPD normally cooperates with TRPE. Let us see in the figure now. So here we can see, okay, now you can able to see it clearly. So TRPE showing arrow mark, it shows anthranilate synthase. In bracket it says CO1. TRPD encodes anthranilate synthase CO2, or you can say phosphoribosyl anthranilate transferase. Both of them together form the main functional enzyme. Both are the subunits. Together forms the main enzyme anthranilate synthetase. Consists of CO1 and CO2 units. CO1 and CO2 are actually, here it is given very clearly, CO1 and CO2 are the component 1 and component 2 of the enzyme, right? So these are the first two genes which encode only one enzyme, which is anthranilate synthetase, as we can see here. And this anthranilate synthetase derived from anthranilate synthase, okay, which is CO1 component 1, and anthranilate synthase, or you can say phosphoribosyl anthranilate transferase, which is the component 2 of that enzyme. Anthranilate synthase derived from TRPE. Better to say it is coded by the gene TRPE, and anthranilate synthase component 2, which is phosphoribosyl anthranilate transferase, it is coded by the gene TRPD. So TRPE and TRPD together form the enzyme called anthranilate synthetase, right? Your next is TRPC. TRPC encodes the enzyme phosphoribosyl anthranilate isomerase. Phosphoribosyl anthranilate isomerase. What does this enzyme do? Let us see in here. Wait for a minute. Here it is given. Okay. So TRPC consists of two domain. TRPC consists of two domain. The name of the two domain is indole 3-glycerol phosphate synthetase, and number 2 N5' phosphoribosyl anthranilate isomerase. So this TRPC consists of two domains. One is your indole 3-glycerol phosphate synthetase, and the second is N5' phosphoribosyl anthranilate isomerase. This TRPC is a bifunctional enzyme. that catalyzes two sequential steps of tryptophan biosynthesis. This TRPC, which encodes this enzyme phosphoribosyl anthranilate isomerase, as you can see here, this enzyme is a bifunctional enzyme. It catalyzes at least two steps needed for tryptophan biosynthesis pathway. The first reaction is catalyzed by the isomerase domain, as I have written. The two domains N5' phosphoribosyl anthranilate isomerase, it catalyzes the first reaction coded by the TRPF domain, and the second reaction is catalyzed by the synthetase coded by the TRPC domain. So it will catalyze the two reaction needed for the tryptophan biosynthesis. Okay, so let us see in the figure our TRPC. Okay, TRPC encodes this one, phosphoribosyl anthranilate isomerase. A comma has been given, indole glycerol phosphate synthetase, because it has two domains, right? It has two domains and it catalyzes two steps. It catalyzes two steps in the tryptophan operon in the tryptophan biosynthesis pathway, right? Okay, so again I repeat, this TRPC consists of two domains. One is indole 3 glycerol phosphate synthase, and another domain is called N5' phosphoribosyl anthranilate isomerase. Normally TRPC is a bifunctional enzyme. TRPC, the enzyme that encoded by TRPC, is a bifunctional enzyme. It catalyzes two reactions for the tryptophan biosynthesis. First reaction will be catalyzed by the isomerase domain, and a second reaction will be catalyzed by the synthetase domain. Okay, then comes the last two enzyme, which is your TRPA and TRPB. These two genes will encode only one enzyme. TRPA and TRPB are the two subunits of tryptophan synthetase. TRPB and TRPA are the two subunits of tryptophan synthetase. It combines with TRPC product with serine to produce tryptophan. Again I repeat, TRPA wait TRPA and TRPB are the two subunits of tryptophan synthetase. This TRPA and B form this tryptophan synthetase and it combines with TRPC's product with serine to produce the tryptophan. So here we can see TRPB and A. As you can see here, TRPB encodes tryptophan synthetase beta and TRPA encodes tryptophan synthetase alpha. The two subunits combine to form tryptophan synthetase alpha 2 beta 2, and it catalyzes it actually, with the help of serine, it combines with this phosphoribosyl anthranilate isomerase indole glycerol phosphate synthetase, which are the two domains of the TRPC, to form tryptophan. Okay, so here we can see about the all enzymes, the genes which which are structural genes that produce the tryptophan biosynthesis. Normally here the written material is not too much in a good way. I have written in a bad way. So the tryptophan operon of E. coli controls the biosynthesis of tryptophan in the cell from initial precursor called chlorismate. Okay, and this tryptophan operon contain genes It contains genes for the production of 3 enzymes, right? 5 polypeptide chain produce 3 enzymes. So the tryptophan operon of E. coli controls the biosynthesis of tryptophan in the cell from the initial precursor chlorismate, and this tryptophan operon consists of 5 genes. These 5 genes will encode 3 enzymes which will deals for the production of biosynthesis of tryptophan. Now the product of the E and D, the product of E and D produce the enzyme called anthranilate synthetase. The product of E and D produce the enzyme anthranilate synthetase. This enzyme anthranilate synthetase catalyze the first two reaction in the tryptophan operon pathway, in the tryptophan biosynthesis pathway. So the product TRPE and TRPD genes, the product of TRPE and TRPD genes produce the enzyme anthranilate synthetase. This enzyme anthranilate synthetase catalyzes the first two reaction in the tryptophan pathway. Next enzyme which is responsible for catalyzing the next two step is the in the pathway is your indole glycerol phosphate synthetase, and its product is your found in the C locus, that is TRPC. Within the TRPC gene, within the locus of TRPC gene, there is a domain called indole glycerol phosphate synthetase. Because I have already mentioned here that TRPC contains two domains, two important regions. TRPC contains two important domains. One is called indole 3 glycerol phosphate synthetase domain, and another one is called N5' phosphoribosyl anthranilate isomerase domain. So after the first two reactions, as catalyzed by anthranilate synthetase, next enzyme will be catalyzed by a component, a domain found in the TRPC is your indole glycerol phosphate synthetase, and it is a product of TAC locus. That means within the locus of TRPC gene, this indole glycerol phosphate synthetase domain contain. The last step, the final step in the reaction in the pathway produce tryptophan from indole glycerol phosphate. See what is written? The final step in the reaction is the pathway produces tryptophan from indole glycerol phosphate and serine. So the final step in the reaction is in the pathway it produces tryptophan from indole glycerol phosphate and serine, and this single step will be catalyzed by tryptophan synthetase. Tryptophan synthetase is made up of two proteins, that is from TRPA and TRPB genes. So these the overall about the genes related to the tryptophan operon. We can see TRPE and D, they form the what? TRPE and TRPD are the subunits for the anthranilate synthetase, catalyzing the first two reaction. From chlorismate it prepare phosphoribosyl anthranilate. Then comes the role of TRPC. TRPC is phosphoribosyl anthranilate isomerase and indole glycerol phosphate synthetase. These are the two important domains found in TRPC and catalyze the next two reactions. Okay, next two reaction means it will catalyze the phosphoribosyl anthranilate to convert into INGP, that means indole 3 glycerol phosphate. In the last step, this indole 3 glycerol phosphate will be converted into tryptophan by your tryptophan synthetase. Tryptophan synthetase derived from the two subunits, one tryptophan synthetase beta and another tryptophan synthetase alpha. Both of them combine together to form this tryptophan synthetase enzyme, which convert this indole 3 glycerol phosphate into tryptophan, and for this they require serine for its activity. So this was the main role of the genes which will do biosynthesis of the tryptophan. Now I will talk about the attenuation. Okay. Now what does attenuation means? I have said during the introduction, right? During the just the first page that I had said. What does it say? Let us see. Let us study here. It says attenuation is a mechanism for reducing the expression of the tryptophan operon when the level of tryptophan are high. Attenuation is a mechanism for reducing the expression of the tryptophan operon. It will not stop the expression of tryptophan operon, but it will reduce the expression of the tryptophan operon when the level of tryptophan are high. However, rather than blocking the initiation of transcription, attenuation prevents completion of the transcription. However, rather than blocking the initiation of transcription, attenuation will prevent the completion of transcription. It will prematurely stop the RNA polymerase from transcribing the TRP operon. So, a truncated polypeptide will be made during attenuation if the level of tryptophan is quite high. So, how it is made, we have to see now. Let us see. To understand the attenuation, let's zoom onto a region of the tryptophan operon that we have skimmed over the sections above. We have skipped some of the regions in the tryptophan operon as we have seen in the previous diagram. You can see here clearly. More regions are available in this tryptophan operon which deals with attenuation. So let us see. This is the figure. Here we can see TRPR, which will produce a regulatory protein, the repressor, and tryptophan acts as a co-repressor. When the level of tryptophan is high, this tryptophan will bind to the repressor, change the shape of the repressor so that it can fit into the operator, which is given as the symbol as O. Now next to the operator, better to say between the operator and the genes TRPE, there is a region called TRPL. TRPL is actually the leader sequence. If I zoom it now, you can see here TRPL is the leader sequence, and within this leader sequence there is a region called attenuator. So here is the main function done by attenuation process. When the level of tryptophan is quite high, RNA polymerase will synthesize only this part. It will not move forward. So prematurely the RNA synthesis will stop. So how it do that? let us see. So it is what they are saying. It is saying that there is a sequence that lies between the operator and the first gene of the TRP operon. So between the operator and the first gene of the TRP operon, that is your TRPE, there is a sequence, and that sequence is called the leader element, as we have seen in the figure now, just now. Leader element encodes a soft polypeptide. It encodes a short polypeptide, a short chain of amino acids, and also it contains an attenuator sequence. This attenuator do not code for any polypeptide. This attenuator do not encode any polypeptide, but when it is transcribed into mRNA, it has self-complementary section that can form various hairpin structure. Again I repeat, very important part. Within the leader sequence, I have said about leader sequence that leader sequence is located between the operator and the first gene of the TRP operon, that is your TRPE. So between the between the operator and TRPE gene, there is the leader sequence. Leader sequence encodes a short polypeptide. Within this leader sequence, there is another region called attenuator. Attenuator does not code for protein, but when this attenuator is transcribed into mRNA, it can fold onto itself to form various hairpin loop formation. They have self-complementary sequence, that's why they can able to fold on itself to form various hairpin loop. So attenuator does not encode any polypeptide, but when it is transcribed into mRNA, it has self-complementary sections that can form various hairpin structures. That is attenuator sequence when transcribed into mRNA. It folds onto itself because it has self-complementary sequence to form hairpin loop that can stop the transcription process prematurely. Okay. Here I have written for remembering that it consists of inverted repeat sequence that fold onto itself to form hairpin loop in order to stop the transcription process. Now it should be noted that both in prokaryotes, transcription and translation occur at the same time because they are not enclosed in the nucleus. They are open, they are found openly in the cytoplasm, in the nucleoid, better to say. So once RNA polymerase has started transcribing the operon, on the other hand, the transcribed portion of the mRNA, ribosome can attach to that still forming transcript and begin translating that leader region. So once RNA polymerase has start transcribing the operon, ribosome can attach to the still forming transcript and begin translating the leader region, because the leader region is at the first beginning, right? The polypeptide encoded by the leader segment is very short, just 14 amino acid long, and this leader sequence includes two tryptophan amino acid residues to be incorporated, because the leader segment has such codons which will encode tryptophan, two, one after another, two tryptophan amino acids right next to each other. It has that codons. Leader segment, the leader sequence is just short for 14 amino acid long, and within this leader sequence it has such codons for the 2 tryptophan amino acid. So we require 2 tryptophan amino acid when the ribosome will start translating this leader sequence. So this tryptophan amino acid residue is very important. Again I repeat, the polypeptide encoded by the leader segment is very short. It is just 14 amino acid long, and this polypeptide encoded by the leader segment has 2 tryptophan amino acid residues right next to each other. So the ribosome has to incorporate the tryptophan amino acid when it will observe that codon, respected codon that will encode that tryptophan, right? So these tryptophan amino acid residues are very important here. It plays a very important role. Now, if there is plenty tryptophan, suppose there is plenty tryptophan, then ribosome won't have to wait long for a tryptophan carrying tRNA and will rapidly finish the leader polypeptide very quickly. If there is plenty of tryptophan, then ribosome won't have to wait long. It will quickly allow the tRNA charged with tryptophan because the tryptophan is available. So it will quickly allow the tRNA, charged tRNA, to come and bind and carries out the translation very quickly, and it will complete the translation of the leader polypeptide. Now, if there is very little tryptophan, ribosome will stall, it will stop, it will wait at the tryptophan codon, waiting for the tryptophan carrying tRNA, and will be slow to finish the translation of the leader segment. So when there is plenty tryptophan, ribosome quickly allows the tryptophan carrying tRNA to bind and finishes the leader polypeptide. If there is little tryptophan, then ribosome will have to wait. It has to wait at the tryptophan codon, waiting for the tryptophan carrying tRNA, and will be slow to finish the translation of the leader segment. But the main question arises, what does it matter if the ribosome translates the leader sequence quickly or shortly or slowly? It matters. A very important thing will be presented here. Whether the ribosome is translating the leader sequence quickly or slowly, it affects greatly to the tryptophan operon. As mentioned above, leader sequence is followed by an attenuator region, which in its mRNA form can fold onto itself to form a hairpin like structure. Not only one type of hairpin, an attenuator can form different types of hairpin structure. It can form different types of hairpin structure. Okay, so as mentioned above, leader is followed by an attenuator region. Leader is followed by an attenuator region, which in its mRNA form can fold onto itself and form different combinations of hairpin structure. One of the hairpin structure includes a transcription terminator signal, while the other does not end termination. In fact, it prevents the formation of terminator hairpin. Again I repeat, it will look like difficult, but it's very easy. It will look like easy. I have said that leader sequence followed by an attenuator region. which in its mRNA form, this attenuator will fold onto itself to form various type of hairpin structure. One of the hairpin structure have a transcription termination, will act as transcription termination signal, while there is another form of hairpin structure formed by that same attenuator that will not end the termination. Again I repeat, attenuator, when it is transcribed, when this attenuator region, when it is transcribed to form that mRNA, this mRNA attenuator region can fold onto itself to form different types of hairpin. One type of hairpin structure will act as a transcription termination signal, while there is another form of hairpin structure made by the same attenuator which will not end the termination, which will not end the transcription. In fact, it will prevent the formation of termination hairpin. So if the ribosome translates slowly, suppose, means there is no tryptophan present, it will pause, and its pausing will cause the formation of an anti-terminator hairpin. Again I repeat, if ribosome translates slowly, it will pause, and its pausing causes the formation of anti-terminator, means non-terminating hairpin, okay, formed by that same attenuator sequence. This hairpin will prevent the termination of transcription, and it will also prevent the formation of the terminator loop that causes the transcription to stop. Normally we know that the row dependent termination, the row independent termination. In row independent termination, I have said that there is a hairpin loop form which will prevent the RNA polymerase to do transcription further, and it gets separated out. Okay, that loop will be prevented by this anti-terminator formed by the attenuator sequence, and this will occur when the ribosome translates slowly. Okay, so here you can see here clearly that I have seen only the leader sequence. See the leader sequence given by 1 2 3 4. The number 1, the blue region, encodes the leader polypeptide. 2 3 4 here represents the attenuator regions. The segments of the attenuator has been given by 2 3 4. In case of low tryptophan levels, what happen? This 2 3 4 region of attenuator fold onto itself to form anti-terminator hairpin, as you can see in the diagram. This 2 3 4 region forms the anti-terminator hairpin, and this 2 3 4 region is actually a part of the attenuator region. Now at low tryptophan levels, the ribosome will stall at the leader sequence, and as the ribosome stalls at the leader sequence, this attenuator region, they form a hairpin called anti-terminator hairpin made by 2 3 4 region. This anti-terminator hairpin will allow the transcription to continue. It prevents the formation of terminator hairpin. It will prevent that terminator hairpin to form, because terminator hairpin terminates the transcription. Anti-terminator hairpin will prevent the terminator hairpin to form and allows the transcription process to continue. So this indicates that ribosome stalls. That means it does not get the tryptophan. That means what? That means it needs tryptophan. There is a shortage of tryptophan here. That's why this attenuator sequence fold into anti-terminator hairpin and allows the transcription to continue. and if transcription continues, simultaneously the translation is followed behind. Now if ribosome translate quickly, then what happen? If ribosome translate quickly, it will fall off from the mRNA after translating the leader peptide. If ribosome translate quickly, it will fall off from the mRNA after translating the leader peptide. This allows the terminator hairpin and associated hairpin to form the attenuator then form a terminator hairpin. Again I repeat, if the ribosome translate quickly, it means there is enough tryptophan, because 2 tryptophan amino acid has to be incorporated into the leader peptide because it has tryptophan codons. Okay, so it means the tryptophan is present. If the ribosome translate quickly in the leader sequence, it means it has tryptophan available. It will fall off from the mRNA just after translating the leader peptide, and thus it allows the terminator hairpin to form from the attenuator sequence, and the associated hairpin will also form, making the RNA polymerase to stall, to detach, and ending the transcription process. So let us see. The ribosome had quickly, it has been shown that it moves off. The ribosome had quickly finishes the leader peptide and falls off from the mRNA. That's why a short peptide chain has been formed. Thus the attenuator sequence, with the help of their 1, 2 and 3, 4 segment, they fold onto itself to form terminator hairpin. One, in the sense we know it is the part of the leader peptide, so it binds with each other to form a terminator hairpin that will prevent the RNA polymerase to transcribe further and get released from that mRNA. Actually, when ribosome quickly falls off from the mRNA just after translating the leader peptide, it indicates that tryptophan is available. That's why the ribosome quickly can able to transcribe the leader peptide. This acts as a signal. For the attenuator, so that it can form a terminator hairpin, and thus by formation of terminator hairpin, it prevents the RNA polymerase to transcribe it further. Thus it will prevent the transcription of the TRPE and if the associated genes. In this way, this attenuator is regulating the TRP operon. Again I repeat, just a short overview. When the level of tryptophan is high, then what happened? The ribosome had to wait. When the tryptophan level is low, then what happened? The ribosome had to stall. It has to waiting for the tRNA attached with tryptophan to come and bind and carries out the translation. Now as tryptophan level is very very low, it has to wait, and thus this will act as a signal. It causes the formation of a anti-terminator loop, which is formed by the attenuator sequence. When the ribosome translates slowly, it pauses, and due to its pausing, it causes the formation of this anti-terminator loop formed by the, do not say loop, hairpin, you say anti-terminator hairpin formed by the attenuator, and this hairpin will prevent the formation of terminator hairpin loop, and thus it allows the RNA polymerase to do the transcription, to continue the transcription, and carries out the transcription of TRPE, D, C, B, A, followed by the ribosome also. It will translate, and thus it will synthesize the tryptophan. Now if the tryptophan level is quite high, then what will happen? The ribosome will quickly able to translate this leader peptide and falls off. See, it has been written here as if the ribosome translate quickly, it falls off from the mRNA after translating the leader peptide. and this allows the terminator, it acts as a signal now that allows the terminator hairpin to form. Terminator hairpin is actually formed from that same attenuator sequence, okay, same attenuator sequence and associated hairpin to form, making the RNA polymerase to stall and to detach from the mRNA and end the transcription process. So this attenuation mechanism may be complex, but the result is pretty straightforward. You are able to see that it's very complex, look like very complex, but it is very simple, very straightforward. When tryptophan is abundant, ribosome moves quickly along the leader sequence, terminator hairpin will form done by the attenuator and the transcription process will stop. When tryptophan is scarce, that means low, ribosome will move slowly along the leader sequence, non-terminating hairpin will form, but anti-terminator hairpin will form, and transcription of the tryptophan operon continues. So the logic of this attenuation is same as that of regulation by the repressor of the tryptophan. In both cases, high level of tryptophan in the cell shut down the expression of tryptophan operon. We have seen in the repressor in the first case initially that high level of tryptophan in the cell shut down the expression of the tryptophan operon. Okay, both in the case of attenuation and also in the case of TRP repressor, the regulator gene, the regulatory protein which binds to the operator. Okay, both in both case, high level of tryptophan in the cell shut down the expression of the tryptophan operon. So this was about the tryptophan operon. I hope it will help you by this lecture.